Hey guys, how are you doing? Today we're making another video. I'm gonna go break down various different things. I think that the main objective I'm trying to go for when it comes to explaining what I'm learning is not necessarily have a main topic at hand, but rather just give information on which I'm learning, and so people can follow the same pace at which I'm going. I just actually started editing the past video I made and realized all the mistakes I made in terms of explanations. For example, I think one thing I was talking about was like saying mTOR is a pathway when it's a kinase specifically that gets activated via nutrient abundance and all sorts of things that we'll go over today. But yeah, so when it comes to my videos, just make sure that you don't take it too seriously where it's I'm just reflecting upon my growth, reflecting upon how much I'm progressing on in terms of knowledge and science. So one of my clients is a scientist and a liver scientist, and so I'm going to Seattle in the UW University, and I'll be able to work with him and just ask questions and whatnot, and see what he does for his normal day-to-day job. But just to reassure, don't take any of this seriously. Just think of this as a way as one, this is for me a way to be able to see how much progress I'm making. Even just seeing the last video that happened last week, which I'm editing right now, I'm realizing how much I got wrong and how much I could have explained more in depth. And yeah, in this video, I'm just going to go over some kind of basic ideas that I've been kind of doing my research on, look over notes, and kind of just spew about a little bit and let my ADHD flourish a little. So hope you enjoy it. So another thing to mention before, and so what we're going to really focus on the next thing is the processes for lipids, fats. Last time we really focused on glucose, went through glycogenolysis, gluconeogenesis, glycogen, glycosynthesis. Well, no, glycogenesis and glycolysis, the Krebs cycle, electron transport chain. I was able to connect. Let's get a recap real quick. Gluconeogenesis, for example, is the process of converting, like, for example, amino acids to glycerol, which we're going further. It's actually connecting the dots from, like, triglycerols, right? Triglycerols you break down into free fatty acids, and glycerol gets, and so we actually have like that process in which the glycerol go into gluconeogenesis and converted via free-forming glucose. Then we have glycogenesis, which is the process of converting simply glucose into stored form of glucose. And we have glycogenolysis, which is the process of converting stored form of glucose into moving glucose in the bloodstream, whatnot. And so last time, whether it was detailed enough or not, we kind of just went over the brief explanations upon why these processes are important, why insulin is important via insulin sensitivity for our blood sugar levels. And then we also went over how it could be inhibited, for example, through like the AMPK pathway. AMPK pathway produces insulin. Insulin helps with the process of the cells to absorb glucose, which has a huge impact for example, we can relate to type 1, type 2 diabetes. Type 1 diabetes, they can't even produce insulin, so they have a hard time. Their cells have a hard time insulin sensitivity in absorbing glucose, and the second thing is they don't have a catabolic hormone that can. No, insulin, an anabolic hormone. Forgive me, anabolic hormone that can, and the reason we know it's anabolic is because what it does is it inhibits a catabolic process and helps absorb. And I hope I didn't get that wrong. Yeah, because insulin's also used to, I think, enhance insulin growth factor 1, which is something we'll go way further in terms of a growth factor that helps with, for example, gets attracted via mechanotransduction. Mechanotransduction is a process of, for example, stretching the skeleton has an impact on the extracellular membrane of a cell, extracellular membrane which protects protein cells, for example, and via has a connection with hypertrophy via the stretch, mechanical tension. We'll go further than that way later on. Maybe not, actually, we might go over that today. Depends where we go on. But where was I headed at? Oh, the importance that insulin has, for example, on inhibiting the catabolic process, we know it's an anabolic process. An anabolic process inhibits catabolic processes in order for it to perform an anabolic process via saving energy. And so we also know it's an anabolic process because, like I said, anabolic processes not only, for example, try to save energy via ATP, but also builds. So, for example, we'll look at a kinase mTOR, which is really important in terms of lyosome biogenesis and process of basically building muscle via protein synthesis, and that's an anabolic. That's part of it. The anabolic processes to anabolic kinase via protein synthesis via growth, and so catabolic breaks things down. So I think last time, I was kind of confused about, and I can better explain now, is that catabolism isn't just a process of creating fuel, it's a process of breaking things down, and so anabolism is a process of building things up. And so when we have one more than the other, then then that's when you start getting atrophy, abnormal productions of basically anything, and it's important basically to have a balance in order for your body to efficiently perform optimally and maximally to its fullest potential. So, for example, proteins. When we're exercising, for example, we're mainly in a catabolic state. And for example, when we're going through that muscle contraction, we'll have cell death, we'll have the release of amino acids. This is when we're breaking things down. And then we have, for example, mTOR kinase gets activated via an anabolic state, which will help us gain muscle, which help us recover, replenish. Think of anabolism as building. That's why when we use anabolism to inhibit catabolism, it's because we're trying to build in order for us to perform. Think of it as we have our catabolic processes, and we stop these catabolic processes to preserve ATP. ATP is necessary in order for us to go through these processes. They're very, they're very important for the production of breaking down. Think of, for example, stored form of glucose that gets broken down into free form of glucose. What I think it is that when it tends to be in an aerobic state, when you're able to breathe, use oxygen, for example, this is when the electron transport chain, Krebs cycle, can be used via oxygen combining with hydrogen and then electrons via the electron transport chain to combine to make H2O. And then we have, for example, our Krebs cycle that also requires oxygen. That's when I like to think we're in a catabolic state. That's when we can afford the processes of using ATP, since we're not exerting so much force that we can't breathe, in order for us to convert, for example, through glycogenolysis, to have stored form of glucose into free form of glucose. However, there are times, so lipolysis is the breakdown of triglycerols. So triglycerols is our starting, just think of it as your starting object, right? It's stored in adipose tissue, so adipose tissue, fat tissue, into free fatty acids and glycerol. So we start out with triglycerols, and you break it down via free fatty acids and glycerols, which can be used for energy. These can be oxidized for energy, mainly free fatty acids, or used in gluconeogenesis, mainly glycerol. So free fatty acids can be put via directly in the bloodstream, I'm pretty sure. And then glycerol specifically is what gets broken down through gluconeogenesis, which we went through last time, which can be turned into free-forming glucose later on. That's what it's really cool to look at all the systems of fats, carbohydrates, protein, and see how it all connects together. Because you realize later on as I explain things that, for example, what we have is like oxalate that goes into joining in with the glycolysis, causes, for example, acetyl-CoA to go through a constant cycle via the Krebs cycle. And so the more that you realize that these systems aren't happening linearly, but they're happening periodically with each other, it's really cool to see the kind of expansion and kind of the ideas of thinking that you can go on beyond just simply, oh, this energy system happens because of this. So, and I'm also going to go over kind of like an interesting idea to go for you to think about, and what I'm kind of doing my own research upon that would be interesting after we go over some of these fat processes. So these can be oxidized for energy, like I said, mainly free fatty acids, or used in gluconeogenesis, mainly glycerol. Where does lipolysis occur? It happens in adipocytes, Happens in adipocytes, fat cells, in the cytosol, and lipid droplet surface. Lipid droplet surfaces are like intracellular fat storage units made of triglycerides surrounded by a phospholipid monolayer, which we'll go over and how it gets kind of separated. The surface lipid droplet is where key enzymes like hormone-sensitive lipase (HSL) initiate lipolysis. For now, that's like if that's too complex to understand, just know that they are found largely in adipose tissue, fat tissue. Lipolysis gets stimulated by epinephrine, norepinephrine, glucagon, and cortisol. However, just like glycogenolysis, it is inhibited by insulin. So remember, this is a catabolic process. It is inhibited via insulin. Insulin would inhibit it in order for us to, for example, maximally optimize our ATP production in order for us to, for example, go through that muscle contraction. So let's go through the biochemical breakdown. Let's break this down more in depth. The first step, of course, is the hormonal activation, extracellular signaling. Hormones already mentioned, such as epinephrine, glucagon, etc., bind via estrogenic receptors on adipocyte membrane. This activates G proteins, then adenocyclase, which results in the activation of our dear old friend CAMP. Now, this makes sense, as we also know that CAMP initiates glycogenolysis. Glycogenolysis. I just freaking hate saying all this freaking science crap. Sometimes I don't know how to say the word, and it's annoying. What does glycogenolysis do? I'm gonna remind you again. It's activated activated by glucagon or epinephrine, in which it makes our stored form of glucose, called glycogen, into free-moving glucose in our bloodstream. The CAMP pathway uses energy systems to exert energy in times of need, so it only makes sense that the CAMP pathway takes part in the breakdown of triglycerols to expend energy. So as we know, the next step is that CAMP activates pKa, protein kinase A, then begins to phosphorylate paralipin A. Paralipin A is what surrounds the triglycerols. Think of it as like a protector, and so we want to break down this paralipin in order for our process of lipolysis to continue occurring. Paralipin is a structural protein that coats lipid droplets, shielding stored triglycerides from lipases. When pK phosphorylates paralipin 1, it reorganizes and loosens the coating. And so before we go any further, I'm going to quick test you. Is lipolysis a catabolic or an anabolic process? All right. It's a catabolic process. And so then you're like, well, aren't we breaking down energy? Yes, but remember, we're breaking down triglycerols, which is stored in adipose tissue. So remember, when it comes to glycogenolysis, it's a process of breaking down, for example, a stored form of glucose, glycogen, into free form of glucose. Well, then you're like, well, that's free form of energy in our body. That's what we can use to perform energy. Nothing makes sense now. Calm down. Same thing, was confused. But it requires a lot of ATP in order for us to convert this stored form of energy into free form of energy. Hence why it's a catabolic process. So while there's some anabolic processes such as gluconeogenesis, which is the process of bringing down other substrates into free form of glucose, it doesn't require as much ATP, if I don't know if any, and that's what I'll have to go in depth upon. Go and look myself, is that it's not nearly as ATP needy as, for example, glycogenolysis and lipolysis. So remember, the process of catabolism is a process of breaking things down that are big to small, and so that use a lot of energy, that remove us from performing optimally when it comes to performance. And so lipolysis is a catabolic process. Okay. So this opens up, so we were talking about how paralipin 1 gets loosened and kind of opens up, it gets reorganized. So this opens up physical access to the inner lipid core. It also facilitates the docking of coactivated proteins like CG158, which activate lipases further. Think of paralipin like a bubble wrap around lipids. PK lets the scissors in. However, PK not only phosphorylates paralipin 1, but it also phosphorylates hormone-sensitive lipase, which is HSL. HSL and the floats in the cortisol cytosol when inactive. When phosphorylated by pKa, it becomes enzymatically enzymatically active. Damn. It then translocates to the lipid droplet, now accessible thanks to paralipin 1 remodeling. Once pKa opens access and switches HSL on, lipolysis proceeds as a sequential handoff between three enzymes. Adipose triglyceride lipase, ATGL, starts to break down triglyceride, TAG. This uses diglycerol, DAG, plus a free fatty acid. Then you have HSL then continues the process, converting the diglycerol into glycerol and one free fatty acid. So you have your name triglycerol, which gets broken down to glycerol and free fatty acid. Hopefully that's not too hard to remember. This results in the final product of lipolysis being three fatty acids and one glycerol. These free fatty acids are released in the bloodstream and bound to albumin. Albumin for sure comes from the liver. It's a protein that's very vital. That is, I think, it's one of the most produced proteins. They are transported to the muscle, liver, heart, and oxidized for energy. Deoxidation leads to acetyl-CoA, which results in the product of ATP, and the glycerol is transported to the liver and used in gluconeogenesis or glycolysis, which we've gone in depth plenty of times enough. Explain 100 times to the point that your ears are bleeding and probably want to punch me, but it's okay. So, our next process we're going to talk about is beta oxidation. I can't wait to get done with these energy systems. I personally I love the energy systems, but I'm a nerd when it comes to really freaking the cells and, for example, cytoskeleton and the breakdown of the connection of, for example, enzymes, kinase enzymes such as mTOR, but we'll go on that in a second. So I need to hold my breath for a second and continue on talking about VANS. Deoxidation is a catabolic process that breaks down free fatty acids into acetyl-CoA units in the mitochondrial matrix. These acetyl-CoA molecules then enter the Krebs cycle, and the NADH, FADH produce feed into the electron transport chain to generate ATP. Now let's go back to ATP. And let's go a little more in depth. So first, enzyme acetyl-CoA. I was talking to my liver scientist friend, and he's like, "It's acetyl-CoA, not acetyl-CoA." I'm like, I gotta pop off more in terms of my vocabulary. Okay, first enzyme acetyl-CoA synthetase at the cost of 2 ATP will convert free fatty acid into fatty acetyl-CoA plus AMP plus PPI. Don't even forgot the freaking whole name for that. It's okay. This process occurs in the outer mitochondrial membrane. The next step is to bring the long-chain fatty acetyl-CoA across the inner membrane. However, it can't cross directly. Remember, so it can't cross this membrane directly. So what it's going to do is it's going to, before I go in depth because it can be confusing, it's going to transform back to itself. So a malonyl-CoA inhibits CPT1, which prevents fat breakdown during active lipogenesis. This is why then carnitine palmitoyltransferase 1 (CPT1) will convey fatty acid acetyl-CoA into acylcarnitine. Acylcarnitine is then moved to the matrix. The next step is deoxidation proper, which takes place in the mitochondrial matrix. Once fatty acetyl-CoA is inside the matrix, the deoxidation spiral begins. And so this is where acetyl-CoA then becomes back to acetyl-CoA, which is what I was explaining. It converts into one thing to go back into another thing in order for it to go into the mitochondrial matrix. This process removes 2 carbon units per cycle as acetyl-CoA. As 2 units of carbon are used from acetyl-CoA, the product results in electrons harvested as NADH, FADH2, and a shorter acyl-CoA chain. And so remember this acyl-CoA is maybe it doesn't become. I don't know why I said acetyl-CoA. So it becomes acyl-CoA will and we know that it gets broken down via the process of 2 units of carbon being broken down per cycle, and so this will make the acetyl-CoA chain smaller and smaller each time. The shorter acyl-CoA chain returns for another round of the cycle until all carbon units are removed as acetyl-CoA. If that was confusing, then just be smarter. Just think and just process. No, I'm joking. if that was confusing, just think of it as this, right? Acyl-CoA, a long chain, is broken down by 2 units of carbon in the B-oxidation cycle, resulting in the product of acetyl-CoA. The acetyl-CoA then enters the Krebs cycle, which produces NADH and FADH2, later on producing ATP via the electron transport chain. So, probably what I was explaining earlier was confusing. Just know that when it comes to it, acyl-CoA, which is a long chain, gets broken down constantly through. Remember, what we're doing is we're taking 2 carbon units from acyl-CoA each cycle. And so this results in acetyl-CoA as the end product. And so this is when acetyl-CoA enters the Krebs cycle. I think that when I was explaining it, I might have mixed up acyl-CoA with acetyl-CoA. But acetyl-CoA is the end product and goes in the Krebs cycle. And so we not only produce ATP via, not only going to produce ATP via the Krebs cycle, but also produce ATP via the beta oxidation cycle, in which we broke down acyl-CoA, acyl-CoA, so that we can get acetyl-CoA. So it's like killing 2 birds with 1 stone. But the next process we're going to talk about is keogenesis. Keogenesis is the hepatic, liver-only metabolic, and so that's why we have like hepatocytes, which is what makes up the liver, hepatic, liver-only metabolic pathway that synthesizes ketone bodies from excess acetyl-CoA, primarily during times when carbohydrate availability is low and B oxidation is high. And so before we go into this is what really cool, and this is when you start really connecting the dots. If I remember correctly, we have like oxaloacetate, which is important for the Krebs cycle. It'll go for gluconeogenesis in order for it to be turned free-forming glucose. We use ketogenesis. We're in a state that we need energy, right? So our body's doing everything possibly can to perform that energy. So we use other substrates or use things such as oxaloacetate to go into the gluconeogenesis to perform free-forming glucose. However, this lack of free-forming oxalate, which is important for the Krebs cycle, will cause a problem when it comes to acetyl-CoA, I'm pretty sure, leaving the Krebs cycle. Not acetyl-CoA, pyruvate, from pyruvate leaving the Krebs cycle. So this will then cause for an abundance of acetyl-CoA, which you're about to see. It's actually really cool. So your body performs keogenesis during fasting, carbohydrate restriction, or prolonged exercise. Fasting, what happens is that we're low on a carbohydrate intake, for example, our blood sugar levels are low. We need the process of gluconeogenesis. We need these processes that will increase our blood sugar levels. This is when you're starting to connect the dots. Why does it happen? When glucose is low, insulin is low, triggering lipolysis. Free fatty acid floods the liver, undergoing B-oxidation and resulting in lots of acetyl-CoA. Now, it might get a little confusing, so just pay attention here. During fasting or prolonged exercise, blood glucose drops. The body needs to maintain blood glucose levels for the brain, which prefers glucose, red blood cells, which I was trying to talk about earlier, but I forgot which needed glucose requirement, which requires glucose, kidneys, and testes. The liver must start making glucose from non-carb sources in gluconeogenesis. Due to this, pyruvate from lactate, alanine, or amino acids is converted in oxalate by pyruvate carboxylase, OAA. OAA toward glucose synthesis, gluconeogenesis. Due to OAA going to gluconeogenesis, the Krebs cycle slows down. Pyruvate can either do 2 things: turn into acetyl-CoA via pyruvate dehydrogenase, or enters the Krebs cycle by combining with oxalatate. Now, what do we know happened to oxaloacetate? Oxaloacetate already left to go into gluconeogenesis, which is what I was explaining a second ago, which we need oxaloacetate for pyruvate. So now that oxaloacetate's gone, so as I explained a second ago, we need oxaloacetate goes into gluconeogenesis. This is for us to turn substrates into free-forming glucose. And so in order for pyruvate to turn into acetyl-CoA for the Krebs cycle, it must combine with oxaloacetate. So now that dear best friend is gone, what happens is that this whole process of what would originally be from glycolysis, Krebs cycle, electron transport chain is kind of miscued a little. And so this what ends up causing is for us to be stuck in the Krebs cycle. Now, is this bad? No. What happens is that since we're stuck in the Krebs cycle, what results in ketogenesis is the process of making a whole bunch of acetyl-CoAs. So the reason why that we in ketogenesis we go through and make through a whole bunch of acetyl-CoAs is because we're missing one of the products needed in order for us to go beyond the Krebs cycle, and this is because pyruvate is missing its best friend oxaloacetate. I'm pretty sure. This is because normally OA from what let's read and let's see if I'm right. This is because normally OA from what used to be with pyruvate combines with acetyl-CoA in the Krebs cycle. But during gluconeogenesis, OA is siphoned off. Since it is siphoned off, acetyl-CoA has nothing to combine with, resulting in a buildup of acetyl-CoA, which triggers ketogenesis as an alternate outlet. So where does ketogenesis occur? It occurs in the liver in the mitochondria of hepatocytes, which are cells in the cells that build up the liver, which are the main functional cells of the liver. I just said that. While the liver produces ketones, it does not use them as it lacks thorphorase enzyme. Right. However, tissue that do use it are the brain, heart, skeletal muscles, and kidneys. And so let's go step by step from acetyl-CoA to ketone bodies. The first step is the condensation of 2-acetyl-CoA because the enzyme thiolase. Right, so then a third acetyl-CoA joins, producing 3-hydroxy-3-methyl glutyl-CoA, HMG-CoA. This molecule also plays a role in cholesterol synthesis, but in ketogenesis, it's routed towards ketone body formation. Then HMG-CoA lyase cleaves HMG-CoA to release acetylacetate. Damn words. A true ketone body, and one acetyl-CoA that returns to the pool. So now the liver has produced a ketone precursor, which has two potential fates. The first fate is acetylacetate losing CO2 and becoming acetone. CO2 is not metabolized, but instead exhaled via the lungs, which is where you get the saying "fruity breath," and it comes from in diabetic ketoacidosis. The second fate is the reduction of acetylacetate to B-hydroxybutyrate (BHB), which requires NADH and the enzyme B-hydroxybutyrate dehydrogenase. This reaction is reversible and redox sensitive. If NADH is high, as in fasting, the reaction favors BHB. If NAD plus is high, it shifts towards ACAC, acetoacetate. Acetone. No, acetoacetate. Yeah, but no, yeah, I'm right. Acetoacetate. These two, acetoacetate and BHB, are the real energy carriers. They leave the liver into the bloodstream and are taken up by the brain, muscle, and other tissues, where they're converted back to acetyl-CoA for ATP production. Next thing we have to go through is ketolysis. Ketolysis is the breakdown of ketone bodies, mainly BHB and ACAC, back into acetyl-CoA and non-hepatic tissues. The liver produces ketones during fasting or low carbohydrate intake. Tissues like muscle and brain consume ketones for energy when glucose is limited. Remember, the liver makes the ketones but doesn't consume them. It preserves them for tissues. We will follow BHB because it's the main ketone fuel. The first step is the enzyme B-hydroxybutyrate dehydrogenase, converting BHB into ACAC. It's important to note B-hydroxybutyrate dehydrogenase requires NAD+, making the step redox sensitive. Enter converting BHB and ACAC depending on the cellular NADH-NAD+ ratio, which we went through a second ago. Then the enzyme I don't know how to say this. Succinyl-CoA acetate CoA transferase SCOT, which is a key enzyme absent in the liver, uses succinyl-CoA from the Krebs cycle and transfers CoA to ACAC. The enzyme thiolase then brings 2-acetyl-CoA molecules into the Krebs cycle to produce ATP. At the end of the day, for each ketone, the cell gains 1 NADH from redox step and 2-acetyl-CoA, which result in 20 ATP total via Krebs cycle and electron transport chain. Okay, so now we're going to go over processes of proteolysis, processes of proteins, amino acids. This is going to be really important for you if you're really interested in kind of the process of, how many times can I say process? If you're really interested in the way of how we gain muscle, remember we have to break down muscles in order for us to kind of think of it as ways not regenerate, but for growth, right? And so this is where like proteolysis, the butyrate lysosome system come in, where we're breaking down proteins in order for us to then release amino acids. Then through this process, we end up creating via mTOR, via protein synthesis. We use these amino acids in order for us to gain muscle. And this gets activated via the stretch of the cytoskeleton, via mechanical transduction, the stretch, mechanical tension. But it all starts with the breakdown of proteins and then protein synthesis. So proteolysis is the enzymatic cleavage of peptide bonds, breaking proteins into smaller polypeptides or free amino acids. It happens in several contexts, such as replacing damaged or old proteins, providing substrates for energies for gluconeogenesis, activating complement proteins, processing antigens, and activating precursors. There are also different types of proteolysis along with different jobs. Proteolysis is harmful when it exceeds the body's ability to be rebuilt. Remember, as I went through earlier, there's a process of being too much in an anabolic and catabolic state. Anabolism is the way to build. Catabolic breaks down. This is catabolism, by which we're breaking down proteins in order for us to release amino acids and then hopefully replenish with being in an anabolic state. However, if we're constantly in a catabolic state, then we're constantly breaking down. We're not allowing ourselves to replenish, and that's where we come with atrophy and sarcopenia and much more. So it is important to find a balance between this anabolic and catabolic processes, by which you're providing enough stress to the muscles while also not providing too much stress. They're not allowing yourself to recover. However, you need yourself to provide stress in order for you to then need to recover and build, which is where we'll go later on, and it comes to the necessary requirement for us to be able to get in that stretch position, to constantly causing stress in order for us to not lose muscle. However, our cells over time become less flexible via the ECM, extracellular matrix, and so this has a huge impact towards why older people have a harder time gaining muscle. It's because it's not as flexible. It's harder for mechanical tension, mechanical transduction, the breakdown of connection of proteolysis. And so you realize that the more you kind of go more in depth, the more all this connects into one huge system that's not only simple but very complex. So there are also different types of proteolysis along different jobs. Proteolysis is harmful when it exceeds the body's ability to rebuild. It leads to muscle wasting, organ damage, immune suppression, and toxic byproducts, especially in chronic illnesses, starvation, or catabolic states. Think of it like this. Proteolysis in general is supposed to allow for recycling by breaking down proteins into small amino acids for key pathways within proteolysis. However, excess proteolysis leads to too much breakdown, not allowing the body to catch up and recycling these amino acids in a healthy way, and causes too much energy drainage. It's like doing cardio. A good amount of cardio is good. However, too much can lead to atrophy and overtraining. That's why AMPK pathway inhibits proteolysis via mTOR, because mTOR is a kinase that is pro-muscle growth, and which inhibits proteolysis and allows the process of protein synthesis to occur rather than protein breakdown, in order to accumulate strength and allow myofibril tissue growth via myocytes, sarcomere, myosin, actin, whatnot. So we're briefly going to go over the key pathways and enzymes. There's the proteasomal pathway. The ubiquitin proteasome system tags proteins with ubiquitin and degrades them in the proteasome. This process is ATP dependent, selective, and handles short-lived or damaged proteins. The system is crucial in controlling transcription factors, cell cycle regulators, and metabolic enzymes. It breaks down proteins into amino acids to either be recycled or for energy usage, or undergo a process to release ammonia from protein, which is then excreted via the urinary system. We'll kind of go in depth upon how we have like NH3, ammonia, which goes through the urinary system, which has a connection with your kidneys. You have your adrenal glands, then you got your kidneys, and then underneath your kidneys, you have your urinary system where you piss. Think of ubiquitin as a molecular sticky note that says degrade me. The E1 enzyme activates ubiquitin. Next, E2 and E3 enzymes help transfer ubiquitin to target protein. Often, polyubiquitin chains are attached for clear recognition. This tagging doesn't degrade the protein, yet it's marking for it for breakdown. Now, the tagged protein heads to the 26S proteasome, a large barrel-shaped enzyme complex. Inside the proteasome, the protein is unfolded using ATP, fed into catalytic core, and peptide bonds are hydrolyzed, chopping the protein into short peptides, 7 to 10 amino acids. These peptides can be sent to the cytosol for further trimming into free amino acids or directed into the antigen presentation pathway, such as into MHC1 in immune cells. Next, each amino acid can now go into two directions: nitrogen management via transamination to deamination, which will go in a second, which leads to the urea cycle, or carbon lyosome pathway, autophagy, is when organelles and proteins are engulfed in autophagosomes, a spherical structure with double layer membranes in a cell that help with clearing out damaged part of our cells, which get digested by lysosomes, membrane-bound organelles that act as the cell recycling center, digesting waste, cellular debris, and foreign invaders to maintain health. This pathway gets activated in nutrient deprivation or NPK signaling. It handles bulkier targets, not just individual proteins. Remember, NPK pathway can only get activated via the process of, for example, stress or going through exercise, but it also gets activated via nutrient deprivation, while catabolic processes get activated via nutrient abundance. So the first process we're going to go through is transamination. Transamination, let's say alanine arrives in the liver, it comes from muscle carrying oxygen. Alanine wants to go to the urea cycle because it's a safe way to dispose of toxic nitrogen. When muscle breaks down protein during exercise or fasting, amino acids like glutamate pick up nitrogen groups from alanine. Alanine is formed from pyruvate via the enzyme alanine aminotransferase, ALT, which uses coenzyme pyridoxal phosphate, PLP, derived from vitamin 6. In this reaction, alanine donates its amino group to a ketoglutarate, forming pyruvate and glutamate. Glutamate now acts as the primary nitrogen carrier. It temporarily holds the nitrogen before transporting it into the liver mitochondria, where it can release the nitrogen as free amino via glutamate dehydrogenase. This ammonia then enters the urea cycle for safe excretion. Thus, while a ketoglutarate receives the nitrogen, it is the glutamate that carries towards the urea cycle. Basically, pyruvate turns into alanine, and in turn back into pyruvate once alanine releases nitrogen and gives it to glutamate. The leftover pyruvate at the end gets used for gluconeogenesis, while the glutamate goes on to the next step. So now we're going to go into deamination, which is the caspase mediated pathway. This pathway is specialized for apoptosis, which is cell death, and cell dismantling. Remember, apoptosis is a process that is controlled, while we have like this process called necrosis, which is an uncontrolled cell death. This is where we have inflammation, and I think we go over it in a little bit, but apoptosis natural is good, necrosis not. Deamination, the nitrogen removal. Inside the liver, glutamate now moves into the mitochondria, where the enzyme glutamate dehydrogenase, GHD, waits. The cell is under metabolic stress, let's say low ATP and high ADP, high ADP, GDH gets activated. GDH strips the amino group of glutamate entirely, releasing it as free ammonia, NH3. The leftover carbon skeleton, a ketoglutarate, and re-enters the TCA cycle, and nothing goes to waste. Now you've got ammonia flowing inside the mitochondria, toxic but ready to be packaged. The liver then takes NH3 and combines it with CO2 and ATP to make carbonyl phosphate. I don't know. Then you take it to the urea cycle. The urea cycle is part of the urinary system. The cycle safely seals 200 atoms into the molecule of urea, which then exits into the blood, travels to the kidneys. Okay, so not in the into the urinary system, but it goes to the urinary system in order for it to be excreted, travels to the kidneys, and gets filtrated out into the urine. In short, transamination moves nitrogen from glutamate. That nitrogen becomes ammonia, and ammonia enters the urea cycle, safe for excretion. Remember, our kidneys specifically not only get, for example, regulated via aldosterone, via I know it gets stimulated via aldosterone. Aldosterone actually increases, I'm pretty sure, volume within the kidneys. but kidneys is really important when it comes to, for example, sodium intake, when it comes to our electrolytes, comes to urinary release and whatnot, which is why the urea cycle and the release of ammonia leads to the kidneys, which then leads into the release of the urinary cycle via urethra. Okay. So the regulation of proteolysis. Insulin suppresses proteolysis as it promotes anabolic metabolism. Glucagon or cortisol stimulates proteolysis for glucose production. AMPK activates boosts autophagy, which is a process of breaking down cells. We can connect this to how stress cortisol ignites growth, which is activated via AMPK and via mTOR C1 through mTOR through the use of extracellular signaling, promoting proteolysis and protein removal and production. But we'll talk about that later. mTOR signaling inhibits autophagy and proteasomal degradation during nutrient abundance. When we have nutrient high abundance energy, we don't necessarily need AMPK, since we use that as a precursor for making more energy. Instead, we will go through CAMP, which uses high energy as fuel. Okay, so we're going to go more out there. So we just talked about, for example, lipolysis, proteolysis. We're going to continue on with like protein breakdown, but for right now, we're going to go in a bit of a leeway. We're going to talk about cytokines, okay? So what the hell are cytokines? Cytokines are small proteins released by immune cells and some non-immune cells to communicate and orchestrate responses. Think of them as common signals for inflammation, cell death, growth, or repair. Remember, you can have inflammatory cytokines, and then you can have pro-inflammatory cytokines and the anti-inflammatory cytokines. Some pro-inflammatory cytokines, TNF-A, which is secreted by macrophages, a type of white blood cell that plays an important role in the human immune system and carries out various functions, including engulfing and digesting microorganisms, clearing out debris and dead cells, and stimulating other cells in immune function. They are a defense against antigens. T cells, a type of white blood cell called lymphocytes, they help your immune system fight germs and protect you from diseases. It mainly functions for fevers and secreted by macrophages, adipocytes, and myocytes. Its main function is inflammation, acute phase response, and metabolism. Okay, IFNY is another one, and it's very similar in the other two. So as you can see, cytokines are a huge factor when it comes to like immune system protection. Some are pro-inflammatory, some are anti-inflammatory. They're vital in terms of overall like diseases, for example, IBD, and they're important for metabolic function, which interferes with our energy systems and producing energy, etc. So let's go a little bit on like cytokine metabolic crosstalk. Adipose tissue becomes inflamed, and macrophages and adipocytes release TNF-A and IL-6. Right, so these cytokines interfere with insulin receptor function and glucose uptake, and are contributing to insulin resistance. IFNY further polarizes macrophages to the M1 pro-inflammatory type. So let's just look over some pro-inflammatory and anti-inflammatory cytokines. Pro-inflammatory cytokines promote immune activation, fever, and inflammation. They're secreted by macrophages, M1, Th1, dendritic cells, NK cells. Pro-inflammatory examples: TNF-A, IL-1B, IL-6, IL-12, and it goes on. Now let's look at some anti-inflammatory cytokines. Anti-inflammatory examples: IL-10, IL-4, IL-13, TGFB. You really don't need to like obviously memorize all these cytokines because there's freaking a crap ton. But just know that these cytokines are vital in terms of immune function. They're released by immune cells, and they're small proteins. Okay, so now the next thing we're going to go into is apoptosis. And this is what we'll kind of connect apoptosis and then necrosis is cell death. And it's really important when it comes to proteolysis. Apoptosis is programmed cell death, a highly regulated, energy-dependent process that cells use to self-destruct when they're no longer in need or potentially dangerous. Is non-inflammatory, unlike necrosis, which we will go talk about in a bit. Apoptosis is crucial for tissue homeostasis, immune development, and eliminating damaged cells. Think of apoptosis as a self-contained demolition of a cell, where the building carefully takes itself apart brick by brick without damaging structures. There are two main pathways: intrinsic via the mitochondrial pathway and extrinsic via the death receptor pathway. So we're first going to go through the intrinsic pathway. The intrinsic pathway is triggered via DNA damage or oxidative stress. Oxidative stress occurs when there's an excess of free radical unstable molecules that can damage cells and not enough antioxidants to neutralize them. Remember, so when we go through AMPK pathway, we actually increase our oxidative stress. And so as we increase our oxidative stress, this causes the intrinsic pathway of apoptosis to occur. And so we can use this as a reminder that the AMPK pathway is the process of breaking, for example, of cell death, breaking of muscle cells. That can be a bit confusing because when we think about catabolism and anabolism, I said anabolism is the process of creating, catabolism is the process of breaking down. However, in this case, with anabolism, right, we're breaking down muscle cells in terms of things such as oxidative stress, which get increased via the AMPK sensor pathway, and so this is what causes apoptosis to occur, which will allow us to then be at a catabolic state later on to then have protein synthesis. An anabolic state via protein synthesis. However, the AMPK pathway inhibits mTOR. So while both processes are anabolic, both processes don't occur at the same time. mTOR gets ignited via energy via nutrient abundance, and so this will help with the creation of new muscles. However, and it's a anabolic enzyme, which is a kinase enzyme, right? However, though, it doesn't get activated while we're in that stress state of breaking down muscles, because there's an anabolic pathway, AMPK pathway, that's igniting the process of breaking down muscle cells. So also realize that while we do have anabolic processes, they don't all occur at the same time, via, for example, breaking down while also creating new ones. The intrinsic pathway is triggered via DNA damage or oxidative stress. Oxidative stress occurs when there's an excess of free radical, unstable molecules that can damage cells and not enough antioxidants to neutralize them. Free radicals are produced naturally during metabolic processes, but external factors like pollution, cigarette smoke, and certain chemicals can increase the levels. When free radicals overwhelm the body's ability to detoxify and repair the damage, oxidative stress results in cellular injury. Oxidative stress is linked to a variety of chronic and degenerative diseases such as cancer. Free radicals can damage DNA, increasing the risk of mutation that leads to cancer, cardiovascular disease, Alzheimer's, neurological disorders, Parkinson's disease, and so much more. The cell stress, such as with DNA damage or oxidative stress, results in activation of BH3 only proteins. These inhibit anti-apoptotic BCL2 family members and activate pro-apoptotic proteins, BAX, BAK, oligomerize, process by which two or more protein molecules associate to form a larger, often functional complex called an oligomer, which is important for structure and activity for many proteins. The cytochrome C is released from the mitochondrial intermembrane space, in which it binds to APF1 in the cytosol and forms apoptosome. The apoptosome recruits and activates caspase 9, which in turn activates executioner caspases that handle dismantling of the cell. And so we have this extrinsic pathway that happens via the externally outside the cell. And so as we continue on, these extrinsic and intrinsic pathways end up meeting with each other either way as we go through that execution phase. The execution phase is the same via extrinsic or the intrinsic pathway. The extrinsic pathway is triggered by external signs via death ligands. FSL, TNF-A, death ligands are extracellular signals that bind to death receptors on cell surfaces, which trigger apoptosis or programmed death of the cell. The ligands will bind to a death receptor, in which the death receptor recruits FADDA, fast associated death domain. When a death signal like FAS ligand binds to a cell's death receptor, it brings in a helper protein called FADA, which grabs and holds pro-caspase 8 together. They form a group called the DISC, death-inducing signaling complex. This activates caspase 8, which then starts killing the cell by turning on caspase 3, the main executioner. In some cells, called type 2 cells, like liver cells, caspase 8 also causes protein called BID, turning it into TBID. which goes into the mitochondria and activates the intrinsic mitochondria pathway. This makes the death signal even stronger by involving the mitochondria, so both the extrinsic and intrinsic pathways work together to ensure the cell dies. Here's what happens in necrosis. There's cell injury, which results in damage to the plasma membrane or organelles. There's ATP depletion, failure of iron pumps, especially and sodium, potassium, ATP base, loss of osmotic balance, which results in cell swelling due to water influx, lysosomal rupture, enzymes and basically the whole cell just breaking apart. Overall, the big difference between necrosis and apoptosis is that apoptosis is both efficient and doesn't cause inflammation, as it doesn't spill out like necrosis. Luckily, we're getting to the exciting part. Now we're going to more into kind of the creation of cells, myogenesis, and also, creation of muscle cells. We're going to go a little bit in depth upon mechanical transduction soon. We're excited. Myogenesis is biological process by which skeletal muscle mass, muscle tissue forms, primarily from mesodermal progenitor cells during embryonic development, and also in adult muscle regeneration via satellite cells. Skeletal muscle stem cells that grow, maintain, and repair muscle tissue. Satellite cells are controlled by mTOR, Notch, WNT, and inflammatory cytokines. So satellite cells are really important. Cytokines have a huge impact towards the immune system, and it has an impact on satellite cells. Satellite cells are affected by mTOR, which we'll go in a second. So think of satellite cells as protein synthesis, pro-myogenesis, because myogenesis is the biological process by which skeletal muscle tissue forms because from mesodermal progenitor cells. Okay, there are two types of myogenesis: embryonic myogenesis that builds initial muscle structure, and adult myogenesis repairs or regenerates muscle after injury or hypertrophy stimuli. So we'll go over myogenesis and focus more in terms of adult myogenesis than embryonic myogenesis. The mesodermal progenitor cell originates from the peroxial mesoderm, specifically somites. Within somites, cells in the dermatome express key transcription factors that direct them to become myogenic progenitors. PX3, PX7 determine commitment to the myogenic lineage. MyF5 and MyoD, myogenic regulator factors, MRPs, initiate the myogenic program. The first step is proliferation of cells. The proliferation is the process of splitting apart and creating more, and then differentiation of cells comes afterwards. Differentiation is a process of a stem cell having a direct responsibility given to do something, such as a stem cell turning into, let's say, an osteoblast. The first step is early myogenesis. Growth factors like FGF keep myoblasts proliferating. Something F growth factor. I forgot what it is. Oh, I think it's fiber growth factor. FGF keep myoblasts proliferating, dividing. When these growth factors decline, the signaling cascade that promotes division, like ERK, MAPK, which is really important, is a pathway that ignites ignition. I haven't studied this one for a bit, but the MAK pathway is when mitogens go into receptors. These, receptors connected to the outside of the cell, extracellular membrane. It then goes in and ignites RAS or whatnot. And so these kinases, I'm pretty sure, go in terms of separate copy. I don't know if it's copying his DNA, but what it does helps with proliferation in terms of making two new daughter cells and helps with proliferation of cells. But I haven't studied that a long time ago, for a bit now, so that's a little off topic. But an environment shift towards differentiation instead of proliferation. However, let's go back and explain the process of proliferation and myoblast. Myoblast is a progenitor cell that, when it stops dividing, enters myogenesis to develop into a myocyte muscle cell and skeletal muscles. So, remember, myogenesis is important for protein synthesis. It is a production of muscle tissue via cells. Right, and so these cells happen through division, and which is called proliferation, and then have turn differentiation, which they become, and then they turn into myocytes, which is, remember, differentiation when they're given a job in order for us to create skeletal muscle tissue. Once committed, the cells become myoblasts, which they proliferate under the influence of growth factors like fibroblast growth factor. They play a significant role in wound healing and tissue regeneration, involving regulating metabolism and energy homeostasis. IGF-1 also plays a role in the proliferation of myoblasts. Once growth factor signaling drops, once growth factor signaling drops and proliferation drops, that's when we go into differentiation. So myoblasts exit the cell cycle, align, and prepare to fuse. MyoD and myogenin are myogenic factors, MRFs, transcription factors that turn on specific genes. MyoD initiates muscle gene expression, while myogenin promotes terminal differentiation and fusion. Myoblasts line up next to each other through coherent-mediated cell adhesion. They reorganize the cytoskeleton and membrane protons to be ready to fuse into the multinucleated myotubes, the precursors to mature cells. These myoblasts fuse to form multinucleated myotubes. Before they're mononucleated, but then they become multinucleated. This fusion is regulated by M-cadherin integrins. Integrins are a part of the extracellular matrix we'll go in terms of epithelial cells and how that protects all these cells from taking in damage or any sort of thing like that. Expressing structural proteins like myosin, heavy chain, MyHC, actin, and desmin. So, remember, okay, so as we go further, just as a brief overall summary of basically what I was explaining, myocytes specifically get proliferated, so they start accumulating, and then after the proliferation ends, then they go through differentiation, which, like I said, like stem cells are given a specific job and function to do, in which they're given the job for the production of sarcomere, for basically muscle cell growth. Not muscle cell growth, like myofibril construction via transcription, via sarcomere production, I think through myosin and actin. And so, example, myogenesis and apoptosis working together. So, for example, like this. So this is our connection with, for example, through cell death and protein synthesis. And that's where I was talking about anabolism and catabolism. You're lifting weights, you stress your muscle, which results in some damaged myocytes that undergo apoptosis due to oxidative stress, which causes apoptosis to occur, and calcium dysregulation, which, remember, calcium is really important also when it comes to the muscle contraction. We need that in terms of causing that muscle to contract. We'll go further than that at some other point. Satellite cells proliferate and differentiate into myoblasts upon activation due to stress. Satellite cells are inactive in healthy muscle tissue, but when they're wounded, for example, not necessarily wounded, or also, so satellite cells are important for, remember, recovery. It comes to satellite cells are important for repair, recovery, and whatnot. So they're inactive during healthy muscle tissue. Doesn't mean when we work out, we recruit satellite cells. And so it doesn't mean the muscle tissue is unhealthy, but unhealthy in the sense that it's damaged via exercise, being a stress state. So satellite cells play a vital role in myogenesis, the process of muscle formation, as it's trying to reform and remake new tissue via construction of myocytes going into, or myoblasts. Is it myocytes or myoblasts? Yeah, myoblasts going into allowing transcription and creating via myosin actin to create sarcomere. The main contractile unit of muscle fiber in the skeletal muscle. And so they can also give rise to other cell types too, such as adipocytes and osteocytes, bone cells, although their primary function is muscle regeneration. So now we're going to go to mTOR, which is kind of what's going to connect everything together. mTOR is the kinase enzyme that allows for more, basically more production, which will be important vital in terms of muscle growth. Okay, mTOR stands for mechanistic target of rapamycin, a serine kinase that acts like the cell's contraction hormone. When resources are abundant, mTOR gives light to build, grow, and store. There are two major complexes, mTORC1, growth metabolism, protein synthesis, lipogenesis, and nutrient sensing, and then there's mTORC2, which we're not going to focus on, cytoskeleton regulation, AKT activation, and cell survival. For now, though, we're going to focus on mTORC1 because it's what has a huge connection when it comes to protein synthesis and basic overall muscle growth. mTORC1 doesn't activate randomly. It needs clear signals such as insulin or insulin growth factor binding to receptor tyrosine kinases, which result in the activation of AKT, protein kinase B. Basically, when we work out, we have these kinases that go onto the cell, which will ignite mTOR to attach onto the lysosomes within our cell. Remember, lysosomes are important for Remember, lysosomes are important when it comes to protein creation within the cells, and so mTORs are actually going on the surface of those lysosomes and enhancing lysomyogenesis and protein synthesis production and whatnot. So mTORC1 doesn't activate randomly. It needs a clear signal such as insulin or insulin growth factor 1 binding to tyrosine kinases. AKT inhibits TSC1/2, a GTP ACE activating complex that normally keeps mTOR off. So remember, these kinases are actually what inhibit other complexes that would overall inhibit the growth of muscle. So we have inhibitory complexes that inhibit mTOR, and that's why working out is vital in terms of muscle growth because it inhibits those pro-inhibitory mTOR complexes that would further reduce muscle growth without it. All right, so this GTPase activating complex keeps mTOR1C off under conditions of low energy, low nutrients, hypoxia, or stress by inactivating Reb, a small GTPase that directly activates mTOR. mTOR C1 integrates signals from nutrients, especially amino acids, growth factors, oxygen, status, energy levels, and peak pathway. So what's really cool about this Reb is that Reb actually gets ignited via insulin, and so insulin is pro-mTOR. So, like I said, mTOR goes into the mTOR's protein synthesis, and insulin not only helps with cells taking in glucose and having our blood sugar levels being at level, but it also ignites mTOR via Reb, RHEB. Right, so nutrients, especially amino acids, growth factors, oxygen status, energy levels, and the AMPK pathway. Okay, so once mTOR is on, it phosphorylates major targets such as SC6K1, which boosts protein synthesis via ribosome biogenesis. 4EBR1 releases EOF4E, which allows cap-dependent translation initiation. We got lipin-1, which stimulates lipid synthesis in liver and adipose. And it keeps going on. And basically, mTOR is what's going to ignite translation and allow for biogenesis to occur. So mechanical signals and amino acid sensing. During and after resistance training, mTOR becomes activated to stimulate muscle protein synthesis and hypertrophy. Here's how it evolves. During exercise, mechanical tension activates phosphatidic and integrin signaling at the muscle membrane. What the hell is that? Phosphatidic acid is a bioactive lipid molecule that acts like a second messenger in cells. During resistance training, enzymes like phospholipase D convert membrane phospholipids into phosphatidic acid. PA directly activates mTOR, a major growth regulator. Phosphatidic acid binds directly to FKB12, representing binding FRB domain of mTOR, which inhibits mTORC1 kinase activity, as it competes with rapamycin, a drug that inhibits mTORC1 by binding to the same domain. How is this activated? Mechanical tension activates POD1, POD2, in which POD turns membrane phospholipids into phosphatidic acid. PA then binds to mTOR on lysosomes. mTOR is recruited on the surface of lysosomes, like I talked about, since they handle protein synthesis, which increases protein synthesis. And now that is when now in terms of muscle growth, we're going pro-muscle growth. So the amino acid sensing allows for the process of turns on mTOR, which will create more protein synthesis. Okay, so now we're going to talk about integrins. Integrins are transmembrane heterodimeric proteins made of one A alpha subunit and one B beta subunit. Together, they form a receptor that links inside the cell cytoskeleton to the outside of the cell, the ECM. Think of them as molecular anchors and mechanical sensors. This brings us to understand that integrin signaling is vital when it comes to communication in terms of mechanical transduction and mechanical tension. ECM proteins like fibronectin, collagen, laminin bind to integrins. This causes integrins to cluster and recruit signaling proteins inside the cell. This results in the activation of focal adhesion kinase, SRC kinase, MAPK, P13K, AKT, and RHOGG bases. This allows the cells to then sense mechanical stress, adhesion, changes in cell positioning. The cell modifies integrins from the inside, changing how well they bind to the ECM, which will help improve efficiency of signaling proteins inside the cell. During mechanical loading, integrins sense tension of the sarcolemma, which activates FAK, which resulting in stimulation of mTOR, leading to the contribution of mechanical transduction as well. So I can go more in depth upon this, but I really want to go more in depth upon just the basic explanation, understanding of it. So we have our protein cells, of course, we have our muscle cells, and then we have our ECM, which think of it as like a complex. It's a barrier that protects our cells. The ECM itself is constructed via protogens, I'm pretty sure, which I'm not I have it in my notes, but I'm not fully, in terms of memorization, understanding of it. All I know that is, I think it's protector cells, but it helps protect and form the ECM from any sort of damage going on within our muscle cells. We then have collagen and other proteins that will help construct this ECM. Within our extracellular matrix, we have integrins, and so integrins are what send signals, and they're in with think they're under in the ECM extracellular matrix, which within they receive signals via, for example, mechanical tension, which will help with the production of mTOR and other bring kinases to the cell, which is pro-protein synthesis. Okay, so integrins are within the extracellular matrix, which is a complex that protects. It's a fibrous complex made up of collagen and other proteins to protect surrounding cells, and allow us to grow via protein synthesis. So let's go a little bit more in depth upon that. Okay, so let's go a little bit more in depth on it, okay? So we know, as I've already explained, that we have muscle protein synthesis, which relies on extracellular matrix signaling and remodeling via, when we're in that stretch position, mechanical tension, plus it requires remodeling because of added weight, because what we're doing is stretching the extracellular matrix and the cytoskeleton via weight, via stress. And so I know, don't want to go in another rabbit hole for a second. I'm just going to explain it really quickly. When you think of muscle damage specifically, when you think of apoptosis and proteolysis and this process of basically releasing amino acids, which get recycled, or either get there's ammonia that gets released through the urinary system, and then amino acids get recycled for protein synthesis. The destruction that causes for apoptosis is the extracellular matrix and the cytoskeleton being stretched via the stretch and via tension given by external load. That's why the eccentric is vital when it comes to growth, not only to avoid any sort of injury to your joints, or have it control the descent to avoid any sort of stretch reflex, which is the point in which we're going down, and then your body naturally wants to go up to avoid any sort of injury given by that. Stretch reflex is important when it comes to, like, for example, sports such as football, but is more hindrance and more higher chance of injury, right? But this happens because of us breaking down and causing damage to the cytoskeleton and the ECM, which overall in term is what is called the damage to the muscle causing growth. That's why mechanical tension is so vital when it comes to, and that stretch is vital when it comes to muscle growth. Okay, right. So satellite cells and immune cell crosstalk. Satellite cells get activated. Remember, is a type. It's a protein that gets activated via damage effect and gets wants to repair. Capillary and epithelial barrier support. We'll go through the epithelial barrier in a second, but is what also hovers over the ECM, and so the epithelial cells actually protect the ECM from taking damage and whatnot. Well, mechanical load sensing, mechanical transduction, growth factor, transport, delivery. So actually, before we go into that, let's go into the epithelial cells for a second. Epithelial cells, basal cells, and myoepithelial cells and tissues. So zooming in on epithelial tissues, glands, epithelial cells sit on a thin sheet of ECM called the basement membrane. Underneath the basement membrane is connective tissue, which is rich in ECM made of fibroblasts. This connective tissue also contains blood vessels, immune cells, and nerves, which rely on the ECM for structure and signals. So, epithelial cells sit on the ECM and protect the extracellular matrix system and allow it to do its job to help cell migration, proliferation, more muscle tissue. Epithelial cells are one of the main types of cells in your body. They line the surfaces and cavities of tissues and organs, such as your skin, intestines, lungs, blood vessels, and glands. Their main jobs are protection, secretion, absorption, and filtration. What's actually really cool is that the more in depth we go upon the endocrine system, as we have our liver, we have these liver lobules, and so these liver lobules are protected. So we have our, for example, outside we have our heart, and so we have our hepatic vein, but then we have our hepatic artery, and we have a portal vein. Portal veins are what bring in deoxygenated blood. Arteries are what bring on oxygenated blood, and henceforth, right? We need oxygenated blood. Deoxygenated blood gets recycled and becomes recycled again via oxygenated blood. Right. So, but via this, right, the portal vein and the hepatic artery go into the heart, not go into the heart, they go into the liver. Yeah. They go into the liver, and they meet up with within this triad, which is I think is called a liver lobule. I think I'm pretty sure. Oh, what the hell? I'm pretty sure it's called liver lobule. Last time I checked, I'm pretty sure we have like 50,000. I don't know if it's oh no, it's a portal triad. It's a portal triad. Let me see actually what I'm talking about. Yeah, portal triads are part of the liver lobules. The liver lobule consists of central vein surrounded by portal triads. And so we have our liver along with our hepatic artery and our portal vein. And so this will end up bringing in oxygenated and deoxygenated blood into the portal triads, which are protected via epithelial cells and whatnot. And you'll see it goes into like the very middle, and then henceforth. But we'll go into that. I don't want to get another rabbit hole. We'll go into that later on. But it's really cool. The epithelial cells, and the more that you start realizing the cells and importance, the more you can go into the endocrine system and see the connections it has, rather than going backwards, which I did from the very beginning. Is like try to memorize the whole endocrine system, try to learn how to. It just was stupid. So we know that basically the epithelial cells are important for protection. We got basal cells, the support and regeneration layer. So basal cells are progenitor or stem-like cells located at the bottom basal side of stratified or pseudo-dystrophied epithelial cells. So basal cells are with the epithelial cells, in which they help with cell proliferation, differentiation, and apoptosis. They're really helping for signaling, and they have integrins and have hematoenzymes that physically link to epithelial cells to the basement membrane of the ECM. So we have basal cells, which are with the epithelial cells to help communicate with our extracellular matrix. And then we have myoepithelial cells, and they're the contractile barrier and ECM regulators. They're really important in contractility for secretion, ECM maintenance and remodeling, tumor suppression, polarity preservation. Epithelial cells act as like a barrier and regulator for tissue beneath them, which includes normal stromal cells like fibroblasts, immune cells, and extracellular matrix ECM. So think of the myoepithelial cells as protectors, while the basal cells are ways that which the ECM communicates with the epithelial cells. So I know that So I know that I already like communicated and explained it, but we still have our extracellular matrix and we have our cytoskeleton, but the epithelial cells specifically, given our basal cells and also our myopithelial cells, are will help communicate. They're part of the extracellular matrix and it will help communicate and protect the extracellular matrix, which is the protector of all these surrounding cells for muscle cell damage, whatnot, right? The extracellular matrix has a huge impact um, towards signaling, but also needs to be protected as well, so that that's where epithelial cells come in. So we're almost at it done here. I still have more notes, but I think that I'm personally almost burnt out. The one more thing I want to look at is the connection on like okay. Well, I can't find it. Okay, I can't find it. But basically, what I want to go over really quickly before I end this freaking long, I'm tired, and I already did my notes and study on this, but obviously explaining and understanding is important. Um, as we age, our cytoskeleton and our ECM becomes less flexible, right? So what did I just explain earlier? That the importance of our extracellular matrix and our cytoskeleton stretching being damaged. So the stretch is caused by the mechanical tension, and the damage is caused by the weight via the connection of needing mechanical tension. I just flipped you off. Mechanical tension and mechanical transduction, which is external load that causes proliferation, differentiation via cellular system processes. So mechanical tension plus mechanical transduction is what causes the ultimate optimal damage for our cytoskeleton and for our extracellular matrix, which is important because it gets flexed and damaged for signaling of mTOR and whatnot. However, as we age, our ECM and cytoskeleton becomes more stiff and loses in strength, right? So what this means basically is that it's essential for it to be stretched in order for us to gain muscle, right? However, as we age, it becomes less flexible, it becomes more stiff, and I'm pretty sure it adapts has a hard time adapting, bringing in kinases and adapting to having, for example, protein synthesis and regenerating. So what does this mean for older people as we age, right? When it becomes more stiff, our processes of one, handling stress, but also being able to go via recover via protein synthesis gets worse because for example, our extracellular matrix and our cytoskeleton gets more stiff. So when it comes to muscle growth, that's why it's harder for older people to gain muscle, is because their quality of the cytoskeleton and the extracellular matrix gets worse, and so these vital components of mechanical transduction and mechanical tension aren't as I guess what I would say as optimal. So I think at the end of the day, I mean, we've gone over kind of the stuff that I've gone over a little bit this week. What I plan on going more is in terms of the biomolecular chemistry, and so that's probably what we'll go over next. Remember, all my notes and everything I explained to you is something that I take of that week, and then I present it and kind of test myself in my understanding. Clearly, I have a lot of stuff that I need to go over, but I appreciate all y'all for watching, and I see what I talk about next. But remember, mechanical tension is important for muscle growth via mTOR. AMPK gets activated via stress of, like for example, going through a muscle and exercise. Catabolic processes has a CAMP pathway. It's important for nutrient abundant states.